Hello, I'm Dr. Aden Eagle, and together with my lab partner Dr. Thi-han, we investigated this age-old question. How does water pH affect the growth rate of Paramecium Aurelia? Species of the genus Paramecium are integral in breaking down detritus and thus reintroducing nutrients back into higher trophic levels in aquatic environments. Because they are so common in environments and easily grown, they are also staple protists for research. Additionally, the protists have been found to consume a certain fungus harmful to humans. Paramecium aurelia, one common species of paramecium found in most aquatic environments, has not, however, been researched to ascertain the optimum pH level in its aquatic environment for survival and reproduction. A more complete understanding of P. aurelia tolerances and ideal environments will allow more effective use in medicines and research, and will aid in our understanding of how the pH change of a body of water will alter P. aurelia's rate of growth and development. We hypothesized the following. If Paramecium aurelia prefer acidic environments, then between colonies of P. aurelia that are placed in environments of differing pH values, those being 4, 7, 10, and unaltered, the colony growing in the acidic water will display the greatest overall growth rate at 700%. These procedures were meant to result in 4 plastic containers filled with 29 milliliters of a lettuce, distilled water, pH adjuster, and Paramecium aurelia solution that were identical in all aspects other than the pH of the water. The solution was meant to contain all resources necessary for Paramecium aurelia survival, exempting water pH. Thus, the independent variable was the pH of the solution in which the colonies of P. aurelia were grown. The dependent variable was the growth rate of each Paramecium aurelia colony over a period of 3 days. First, we equipped latex gloves, safety goggles, and lab coats in case of accidents. Using the digital scale, 100 grams of cleaned iceberg lettuce were measured out. The lettuce was then placed in the pot and torn up in order for it to be fully submerged in 500 milliliters of distilled water. After donning heat-resistant gloves, the hot plate was attached to a power source and set to 86 degrees Celsius. The pot was then placed on top of the hot plate, and a thermometer was used to check that the water in the pot had reached the set temperature. A paper plate was also used as a lid to expedite the heating process. The hot plate was then unplugged while the pot cooled to a safe temperature. Next, the contents of the pot were strained through a coffee filter into a beaker in order to separate the iceberg lettuce from the nutritious liquid. Using a pipette, 10 milliliters of lettuce water were then placed into each of 4 plastic cups. The pipette was then washed out using distilled water and dried. Next, after shaking the sample of Paramecium aurelia until homogeneous, a pipette was used to move 2 milliliters of the sample into each of the 4 plastic cups. Once again, the pipette was washed out using distilled water and dried. Each colony was capped and allowed to culture in indirect sunlight for 4 days. Next came the long, slow, laborious process of carefully adding additional distilled water in either pH up or pH down, drop by drop, using a pipette until both the pH of each plastic cup was at a different desired value, those being 4, 7, 10, and unchanged, and the total volume of the liquid in every plastic cup was equal at 29 milliliters. This ensures that the environments in each plastic cup are identical except for the one single factor being changed, the pH of the water. The colonies were allowed to culture for 1 day. A stereoscope was then prepared for use. After uncapping one plastic cup, a pipette was used to swirl its contents thoroughly for 5 to 10 seconds. 1 drop of the liquid was taken from the center of the plastic cup and placed into the middle of a sterile petri dish. The number of Paramecium aurelia present in the drop was then counted and recorded. Using the pipette, the drop was then returned to its original colony. Both the pipette and the petri dish were then washed out with distilled water and dried. The plastic cup was then capped and returned to its environment of indirect sunlight. This process was repeated for the rest of the plastic cups. As shown, all but one Paramecium Aurelia colony displayed a growth rate other than 0, that being a negative growth rate of about negative 92%. Additionally, 9 of the 12 Paramecium Aurelia colonies measured 0 living Paramecium Aurelia as a baseline measurement. Therefore, we can conclude that factors besides the water pH, such as an osmosis induced by use of distilled water, were the main causes of low populations in Paramecium Aurelia colonies. Thus, our hypothesis can neither be proven nor disproven due to a lack of reliable evidence. This information can be applied to all other situations requiring the culture of Paramecium Aurelia colonies, be it medical or scientific research or simply education in classrooms. Our project serves as a clear anti-example of Paramecium Aurelia cultivation. With greater understanding of the larger amounts of food that must be provided for effective and productive growth of Paramecium, all interested in the study of Paramecium will benefit. Some of the most critical limitations to our experiment included our inability to accurately count Paramecium Aurelia populations. Instead of counting the exact number of Paramecium Aurelia in each colony, we had only the technology capable of counting the amount in one drop of water and thus needed to extrapolate outwards, decreasing the accuracy of our measurements. Additionally, our lack of varied Paramecium Aurelia sources was another important shortcoming. Due to limitations in funding, only 2 colonies of Paramecium Aurelia were able to be purchased from a supplier, resulting in heightened susceptibility to a less representative, or different, sample of the protist, one in which the results of the experiment cannot be generalized to include the entire species. Further research on specific food-related needs of Paramecium Aurelia and pH tolerances using more advanced equipment and precise population counting methods would provide a clearer view of the protist niche, benefiting humanity in the form of possible medicines and a greater sobering understanding of the natural world around us. In the words of Carl Sagan, our posturings, our imagined self-importance, the delusion that we have some privileged position in the universe, are challenged by this point of pale light.